Loud? Okay.
Yes,
So it's biotechnology in animal production. It's a 2 credit unit. That is AMP 4504. The aim of the course is meant to provide you with a good knowledge base for the production of healthy animal and improve livestock in Nigeria. And all of this is aimed at sustainable production of livestock. So you should have that basic understanding as the aim of that course, of the course we'll be going through. And there are several objectives, the learning objectives that we will want you to know. After the completion of this course, AMP 504, you will help you to know, basically the definition of biotechnology, recombinant DNA technology, and genetic engineering. So you have a full understanding of what all of this means. Biotechnology. It is coming from 2 words principally, bio and technology. Technology is aimed at animal production, so that we can have sustainable production of livestock. We will also help you to know what is not just definition, but the application, application of that biotechnology in 4 aspects: animal, feed production, nutrition, and reproduction. We also help you to know and to understand the concept of cloning, the concept of marker-assisted selection. We will also help you to understand the physical and genetic mapping of animals, so that when you know the genetic components, you can effectively achieve sustainable production and crossbreeding that is relevant to increase that animal health, improve Nigerian livestock resource. We will also help you to understand the physiological function of farm animals. So when you understand that physiology, you'll be able to get the best of the animal and help them function properly, so that you can get the best from them. We will also help you to know the uses of DNA tests, for meat quality, for genotype identification, for paternity tests also. Because it's good to know the genetic constitution of a particular animal breed. And without knowing also the disease diagnosis, you will not be able to achieve that sustainable growth. So we will also help you to know the different diseases of animal and how it can impact on the sustainable development. So we will also finally help you to know how to conserve those animal genetic resource, the exudes and the insuits. So these are the things we'll help you to achieve at the end of this course. Okay, having given you the highlights and the objective of the entire course, we'll just go straight on to the first module of the course. And the first module, we will be talking about, like I said, the definition of biotechnology and the recombinant DNA technology and genetic engineering. So we will go into just the definition, and for you to have understanding. Now, let me just give you the background. From time immemorial, people have been harnessing biological processes, to improve their quality of life. And in doing so, they are able to help themselves, they are the first agricultural community. So all biological processes is used to improve the quality of life. That is the essence of, the primitive biotechnology, and it can be stated that about 10,000 years ago, about 600,000 years ago, humans began to tap into biological processes of microorganisms in order to do 5 things. They have been able to make bread, alcohol beverages, cheese, and to preserve their dairy products. So biological industry, has been dominated by genetic engineering, really. And this unique concept had helped, scientists, even in their native circumstance, to improve, the likelihood of production of agricultural products, increasing their efficiency and able to get the best of the animal. So, in essence, biotechnology is the use of biological systems, in such organism, and it is used to advance, the animal itself for agricultural practices that will help sustainable production of the animal. So it's the use of technology. And in attempt to use that technology, it must be focused on sustainable development of that animal. The health of the animal is also very important. It's not just, being able to get the byproduct of the animal itself. So we apply it in nutrition supplements, abiotic stress resistance in industry, in strength fibers, in biofuel, and also healthcare. So the basis of biotechnology is actually, to harness cellular and biomolecular processes to develop technologies and products that can help improve our lives and the health of our planet. Okay? So, in essence, we're saying biotechnology is the use of biological system that is found in animal, and we're going to use that living organism themselves to make technology advancement and adapt those technology to various fields. So now, secondly, when we look at recombinant DNA technology, here we are looking at, bringing 2 molecules of different species, insert it in a host organism to produce a new genetic combination. That's why we call it recombinant DNA technology. So we are joining 2 DNA molecules from 2 different species, and we are inserting it into a host organism. And it's like, bringing a man and a woman, the egg in a woman and the sperm in the man, to produce another being. And so this new being is of value to science and medicine, agriculture, industry. And once it is combined together, it enables researchers to do 5 things to determine its molecular sequence. We're able to study the transcripts. We're able to mutate it highly specific ways and reinsert this thing back to the living organism. And when it is reinsert into this living organism, they are able to produce more. They are more efficient. They are able to adapt to their environments, and also not just adapting to the environment, they are able also to produce, produce their kind that will be more productive. So that is the essence of recombinant DNA technology. By the time there is the combination of 2 different species into one, we are able to have a better genetic composition that is able to produce, more virile, more, I don't, what which word will I use now? Okay, let me use that word. More virile species that is able to, produce on a sustainable level. So the third aspect of, the first understanding of this biotechnology is genetic engineering. Here we have, the artificial manipulation, modification, and recombination of this DNA. So number one is we manipulate it, we modify it, and we recombine it. Three aspects. And so when we do all of these, we're able to modify the organism, population entirely. And in doing so, remember, we are focused on sustainable, don't forget that word, sustainable production of livestock. It's not a livestock that can only, after the first, generation or second generation, they begin to reduce in its performance. So by the time all of this is done, the performance of that animal is enhanced after the genetic engineering of, of that animal. So we do it by three things: by artificial manipulation, modification, and recombination of the DNA, so that so that at the end of the day we have modified the organism or the population of the organism. Okay, so we do all of that, to achieve sustainable production, and we use different techniques. Artificial insemination is used, intraviral fertilization is done, cloning of the animal is done, and also genetic manipulation. So this has taken roots in the 21st, from 20th century really, and has gained ground in the 21st century. And so for many scientists, that are interested in agricultural developments to feed the teeming population, this is the way to go. So in summary, we're saying that biotechnology is a biological system found in animals, and in using them to make technological advancements so that we can achieve sustainable development of agricultural products. And also, we told you about recombinant genes, and in doing so, the species achieve a new genetic combination that is of value to science, to medicine, agriculture and industry. Specific to our own course here, we're looking at its value to agriculture, most importantly, the animal species. Okay, so the third aspect is genetic engineering, artificial manipulation, modification, and recombination of DNA. And once this is done, the entire population can have the third, the fourth, generation that is virile, that is productive, and it can lead to sustainable agricultural development of the animal resource. So the first aspect of the module 1, we will attempt to go through it again next week, so that you can have a better understanding of it. We encourage you to listen to this recording over and over, go through your text, and practice the tutor mark assignment contained in your text. There, we're asking you to define biotechnology and give examples of how it can be used in specific terms. Just get to understand the definition of all of these terms, so that as we proceed subsequently to module 2, we'll be looking at the different tools, of genetic engineering, the cloning, the DNA fingerprints, and bioinformatics. So all of this will be shown to you in the next class we are going to have. Thank you very much for listening. So I hope to see you next week. Thank you.